Hello everyone, welcome back. Today I'm here with the fifth part of this question series, that is HPU Botany Entrance Exam 2022. So in this part I will discuss questions from plant physiology specifically. Let's dive. So the question is, absorption of ions against concentration gradient shows. Our options are diffusion, Donan equilibrium, ion exchange, or all of them. So its right answer is Donan equilibrium. It explains the accumulation of some mineral ions inside the cell against the concentration gradient without the expenditure of metabolic energy. It means it is a part of passive absorption. According to this theory, inside the root cell there are some indiffusible ions, or we can say fixed ions, like we have some macromolecules which loses proton. उनसे जब प्रोटॉन लूज होता है, so they obtain the negative charge, and they are fixed ions these are present in the plasma membrane. Such negatively charged ions on the membrane are called as fixed ions. उन्हें fixed ion कहा जाता है. And the plasma membrane is permeable to other types of cations. So the cell has accumulation of anions as fixed ions. Such a cell can absorb cations from the soil in order to maintain the electrical potential balance. So this theory of Donald equilibrium theory, which explains the balancing of charges in case of mineral ions. So if we talk about this discovery, so this was discovered by Frederick Donan in 1911, and it involves unequal distribution of ions across the two sides of a differentially permeable membrane, given the name Donald equilibrium because it is mainly related to the electron neutrality of the charges, because it balances the charges without expenditure of metabolic energy. So we have some fixed ions in the plasma membrane which are formed due to loss of proton from these macromolecules which are present in the plasma membrane, and they are negatively charged, so they can accumulate the positive charged ions from the soil to make the balance inside the cell. So this is known as Donald equilibrium. If we talk about diffusion, as we know that it is the movement of substances of solid, gas or liquid from the region of its higher concentration to the region of its lower concentration, and we can demonstrate this phenomenon with the help of this experiment. If we put a drop of ink in water, we obtain this solution of blue color after some time, because these particles of ink are in continuous motion, and this force that is known as diffusion force, and these particles are known as diffusing particles, and they move from the region of its higher concentration to the region of its lower concentration. This is diffusion. The next comes ion exchange. This is again a part of passive transport, and this involves the absorption of ions on the root surface of root cell membrane, and it may involve exchange of some ions. For example, we have potassium ion. This potassium ion can be absorbed on the surface of root cells by exchanging it with protons. तो प्रोटॉन को एक्सचेंज करके जो प्लांट सेल है वो पोटेशियम आयन को एब्जॉर्ब करता है. So this involves this phenomenon of ion exchange. If we talk about the negatively charged particles, so it can release OH negative ion into the soil, and it can obtain the negatively charged particle inside the cell. So this is called ion exchange theory. It has two mechanisms. One is contact exchange theory, and second one is carbonic acid exchange theory. The next question is, photorespiration is a process in which our options are CO2 and ATP ignite. Phosphoglyceric acid is produced. Respiratory energy is used to produce ADP, and the last option is light energy is converted into chemical energy. So its right answer is phosphoglyceric acid is produced. If we talk about photorespiration, it is also known as C2 cycle. So Otto Warburg in 1920, there was a scientist, Otto Warburg, was working on the process of photosynthesis in green algae. He observed that photosynthesis was inhibited when oxygen is present in larger quantity in the atmosphere, and several other green plants shows this inhibition in the rate of photosynthesis, and this phenomenon was called as Warburg effect, that is inhibition of photosynthesis by high levels of oxygen. तो जब एटमॉस्फेयर में ऑक्सीजन कंसंट्रेशन इंक्रीज हो जाती है, so rate of photosynthesis decrease हो जाता है, and that phenomenon is known as Warburg effect. And this phenomenon was also observed in C3 plants, but C4 plants are efficient plants, they do not show this phenomenon. As we know that this enzyme that is involved in C3 plants is rubisco, which is the most abundant plant protein on this earth's surface. और ये जो rubisco है, it carries both activities of carboxylation and oxygenation. So when the concentration of oxygen is higher, so it shows oxygenase activity here. This RUBP, that is ribulose 1,5-bisphosphate, it combines with oxygen and it produces two chemical compounds. One is 3-PGA, that is 3-phosphoglyceric acid, and other is a 2-carbon compound that is called 2-phosphoglycolate. So this 2-phosphoglycolate is the first stable product of this glycolate is the first stable product of this cycle, so that's why this is also known as C2 cycle. And this photorespiration process involves 3 cell organelles, namely that is chloroplast, peroxisome, and mitochondria. So initially this inside this chloroplast, as we know that this RUBP, that is ribulose 1,5-bisphosphate, it combines with oxygen in the presence of enzyme rubisco. It will form a 3-carbon compound, that is 3-phosphoglycerate, and a 2-carbon compound that is called 2-phosphoglycolate. And for the metabolism of this glycolate, it is firstly converted into glycolate, which is the first stable compound of this reaction. That's why this is also known as C2 cycle, photorespiration. Then this glycolate will enter inside the peroxisome, because the enzymes which are involved in metabolism of glycolate are not present in chloroplast. And here this glycolate is converted into glyoxylate. It undergoes the process of oxidation, and finally it is converted into glycine by the transamination reaction, and this glycine is transferred to mitochondria. Here it undergoes again transamination reaction, and it will form this serine, which enter back inside the peroxisome. Here it is converted back into the hydroxypyruvate after transamination reaction, and it is finally converted into glycerate by the dehydrogenation process. It undergoes the process of dehydrogenation. Then this glycerate will enter inside the chloroplast and is converted back into 3-phosphoglycerate, and this will enter inside the C3 cycle, that is Calvin cycle, when conditions are normal. So in this way this C2 cycle works, and it was thought to be a wasteful process, but now we know that this is important process in C3 plants, and it prevent the photo-oxidation process. Means जो chloroplast का damage होता है due to photo-oxidation, this is prevented. This photo-oxidation of chloroplast is prevented by this phenomenon, that is C2 cycle, and it also involves the formation of certain amino acids which are metabolized in the formation of certain proteins. So this process is known as photorespiration, and here the phosphoglyceric acid is produced in addition to this 2-phosphoglycolate. So photorespiration in C3 plants occur when rubisco adds oxygen to RUBP, forming 2-phosphoglycolate and this 3-carbon compound, that is 3-phosphoglycerate. तो ये भी बनता है, and it is triggered by high light temperature and low CO2 concentration. जब CO2 concentration कम होती है, despite reducing productivity, it offers photoprotective and redox balancing benefits. जो C4 plants हैं, they are highly evolved plants, and to minimize photorespiration losses and thrive in high temperature environments. So these plants are efficient plants, and they do not undergo this process of photorespiration. Next question is, which of the following delays senescence? As we know that this is an aging process, our options are gibberellin, ABA, that is abscisic acid, then we have indole acetic acid, and cytokinin. As we know that we have these categories of hormones of this gibberellin and indole 3-acetic acid and cytokinin. This comes into the category of growth promoting hormones, means they show positive response, while this is ABA is the growth inhibiting hormone. This is also known as plant stress hormone. इसे plant stress hormone भी कहा जाता है. And this is involved in this senescence process. So its right answer is cytokinin. Cytokinin is involved in this prevention of aging or senescence. And this phenomenon is also known as Richmond-Lang effect. This was observed by Richmond-Lang, that if we put a leaf inside a cytokinin solution, it will stay green for a longer duration, because the cytokinin prevents the process of senescence. So Richmond-Lang effect is the retardation of aging due to application of plant growth regulator cytokinins, and the regulator acts by retarding the rate of destruction of chlorophyll and yellowing of leaves. तो basically जो cytokinin है वो chlorophyll की degradation को stop करता है, और जो yellowing of leaf है उसको भी prevent करता है. That is senescence basically. So that's why this cytokinin is involved in delaying senescence in plants. In addition to this, we have some other hormones which plays very important role in plants. Like we have auxin, which is commonly known as the master hormone, master plant hormone. Auxin means to grow, so it promotes cell elongation, and it is involved in apical dominance. Apical dominance means prevention of lateral growth in the presence of apical bud. तो जो apical bud है उसकी वजह से जो lateral growth है वो नहीं हो पाती है, so that inhibition of lateral growth is known as apical dominance. So that's why we do pruning, जिससे हम apical bud को remove करते हैं और auxin की supply को inhibit करते हैं. Then it also regulates phototropism and gravitropism. Auxin is involved in phototropism, and this gravitropism, it stimulates root initiation. Again, this is very important. There is one type of auxin that is known as IBA, that is indole butyric acid. This is mainly involved used for inducing rooting in plants, stem cutting basically. Then this promotes fruit set and growth in certain plants. It is also involved in inducing flowering in plants like pineapple. तो pineapple में ये flowers भी produce करता है. And the bioassay for auxin is Avena coleoptile curvature test. This was firstly observed by Charles Darwin and his son Francis Darwin, and they worked on the grass that is known as canary grass. Next hormone is gibberellin. This is again important plant growth regulating hormone. It stimulates cell division and elongation. Basically, it is mainly involved in cell elongation and breaks seed dormancy. So ये जो है hormone ये involved होता है seed dormancy को break करने के लिए. The next is speed germination. Next is cytokinin. One more important process that is carried out by gibberellin is bolting. Bolting means elongation of stem in case of rosette plants. Rosette plant means which have reduced stem. तो उन plants में ये elongate करता है stem को और flower induce करता है. That process is known as bolting. In addition to this, it also it can also induce the effect of vernalization in certain plants. The next, cytokinin. Cytokinin, it is mainly involved in cell division and differentiation. It delays aging and death, that is senescence. This was the question asked. And it regulates the shoot growth and branching. The next is ethylene. This is the gaseous hormone, and it is mainly involved in fruit ripening, promotes leaf abscission, causes epinasty. Epinasty means the upper portion of leaves grows more as compared to the lower portion, so it shows this bending pattern. This is called epinasty, and it also regulates root hair formation under flooding condition. Next is abscisic acid. It is basically known as stress hormone, plant stress hormone. ये water stress के time पे जो stomata close करता है, so it inhibits cell growth and division. Means basically it induces dormancy and promotes leaf drop, means involved in leaf abscission, and helps plant to respond to water stress. So that's why this is known as stress hormone. The next question is Phytochrom is involved in. Our options are photosynthesis, phototropism, photoperiodism, photorespiration. So its right answer is phototropism. As we know that this phytochrom is a chromoprotein which is responsible for many photo responses in the life cycle of plants. और जो phytochroms हैं ये बहुत सारे responses को carry करते हैं, like we have flowering of photoperiod sensitive species. Then it is involved in detiolation processes. Then it is also involved in dynastic leaf movements, photomorphogenesis. It also helps in synthesis of anthocyanins in plants. And this pigment is water-soluble blue-green pigment, and is sensitive to red light and far red light. And it is present in the two forms. One is the PR form, which absorb the light of red region, and the other form is the PFR form, which absorb the light of far red region. And these both forms are interconvertible form, and this is the only biological pigment which is present in two forms, that is PR form and PFR form, and these both forms are interconvertible forms, and the more active form is PFR form as compared to PR form. And if we talk about its levels, so highest phytochrom levels are found in etiolated seedlings. Etiolated seedlings, and as we know that etiolated seedlings are the seedlings which are dark ground seedlings, means जो dark condition में रोके आती है, and they are pale yellowish in color. So उन seedling में सबसे ज़्यादा amount phytochrom का पाया जाता है. And if we talk about its discovery, so it was first discovered by two scientists named as Borthwick and Hendricks. उन्होंने इसको सबसे पहले observe किया था, and they observed it on during the germination of lettuce seed experiment. And they observed that this pigment, which is involved in absorption of red light and far red light. Then if we talk about the differences between PR and PFR form, so it is PR form is blue-green in color, while this PFR form is light green in color. और जो PR form है ये absorb करती है maximum light of red region, that is 660 nanometers, while this PFR form absorbs light of far red region, that is 730 nanometers. And this PR form is inactive form, this is active form. And if we talk about its responses, so this PFPR form induces flowering in short day plants, while this PFR form produces flowers in long day plants. If we talk about its presence, so PR form is found diffuse throughout the cytosol, while this PFR form is found in discrete areas of cytosol. तो ये कुछ restricted areas में present होता है. And mainly this phytochrom is involved in the process of photomorphogenesis. Photomorphogenesis, this is the process which is carried out by this pigment that is called photomorphogenesis, and this bending of plant parts towards light in response to light, that is called phototropism. इसे कहा जाता है phototropism. So if we talk about some mechanism, so this phytochrom A, there is a gene phytochrom A that regulates phototropism in plants, involving reorientation of hypocotyle towards blue light. And this signaling influences the process of regulating the expression of specific genes that are involved in plant response to directional light cues. So this is a molecular process, and it activates certain genes that helps in showing this phenomenon of phototropism. The next question is removal of seed coat and dormant seed by mechanical method is known as. Our options are scarification, separation, stratification, or emasculation. So its right answer is scarification. So the process of removal of hard coat of seeds to break down seed dormancy is known as scarification. So scarification breaks seed dormancy by enhancing seed coat permeability. There are many mechanical methods which include filling, sandpaper rubbing, nicking, or gentle cracking. Then we have thermal scarification, uses hot water treatment to mimic natural processes. Then we have chemical scarification, which involves soaking seeds in acidic or basic solutions, which helps in loosening of the seed coat. And scarification improves germination rates, aiding horticulture and seed propagation. The next we have this process that is called stratification. Again, this is a process which is involved to break down physiological dormancy, और we can say class C dormancy. इसमें जो seeds हैं they are typically these seeds typically require a period of cold, that is 0 to 10 degrees Celsius, or warm, that is greater than 15 degrees Celsius stratification. This involves placing of seed in moist medium like sand, peat, or vermiculture, and storing them in an appropriate temperature for a specific duration, that is often 2 to 4 months. And the combination of moisture and low high temperature triggers biochemical changes that transform complex food substances into simpler form, utilizing by embryo when it resumes growth during germination. So this cold moist treatment can also reduce the sensitivity of both dormant and non-dormant seeds, and resulting in increased rate of uniformity of germination. The next process we have this emasculation. Emasculation is a process of removal of stamens or male part from the flowers to ensure cross-pollination. So this is called emasculation. This was all about for our today's discussion. I hope you will get some idea from this presentation. Thanks for watching. Have a nice time.